Good evening, everyone. My name is Dan Veru, Co-Chairman and Chief Investment Officer of Palisade Capital Management, and welcome to a very special and timely virtual event featuring Dr. Keyvan Mojarrad, Director of Emerging Infectious Diseases Branch of the Walter Reed Army Institute of Research. We are so fortunate to have Dr. Mojarrad as the world begins the process of vaccinating its people and ending the COVID-19 pandemic. Moderating this evening's event is Dawn Brock, Senior Vice President of Healthcare Equity Research and a member of the small cap equity team at Palisade Capital Management. Dawn's insights and views on opportunities within healthcare were critical in helping us to make sound investment decisions as we navigated through unprecedented fear and market weakness in the first half of the year. And now looking forward, analyzing the tremendous opportunities that lie before us. Before I turn the program over to Dawn and Dr. Mojarrad, I'd like to make some comments on the recent strength in the stock market and the rebound in the economy, along with a brief outlook for 2021. To do this, we must first look back to the first quarter of 2020. This past March was not only a period of extreme stock market weakness, but it also marked the end of a business cycle caused by the government's closing of the US economy. This triggered a sharp recession and a fast rise in unemployment, but it also marked a new way of life from the way many of us conduct business, socialize, and consume goods and services. Many of these trends have created new business opportunities for existing companies and for young growth companies seizing on emerging opportunities. By the spring, a combination of fiscal and monetary policy stimulus restarted the economy and set in motion a historic stock market rally. The recession that ended this summer will go down on record as the shortest in history, even though we still have many unemployed people, and we do need to do more stimulus to support them. Though the stock market has staged an impressive recovery, small cap stocks represented by the Russell 2000 index lagged the recovery as they tend to have more economically sensitive companies and fewer new economy growth companies. All that changed in early November when the results of the Pfizer, BioNTech, and Moderna vaccine trials came out with stunning results of over 90% efficacy. Since those announcements were made, all stock markets are higher, but the Russell 2000, which I have called the reopening of the economy benchmark, has soared over 20%. Clearly, the market believes that these vaccines, and hopefully others that are coming, will be the key to fully reopening our economy. The stock market is looking past the sharply rising infection rates as we should begin to turn the corner now that people are receiving the vaccine. For 2021, growth prospects for the US economy have risen significantly, while interest rates will likely remain low. This remains a favorable combination for stock investors. For most of 2020, technology and healthcare were the dominant sectors, providing most of the returns for investors. Since the vaccines announcements were made, cyclical stocks like industrials, financials, and materials companies have risen sharply, while technology has been flat. As the economy continues to recover, those cyclical industries should continue to do well. Corrections can occur at any time and for any reason, particularly with a new administration taking over on January 20th with different priorities and objectives. But the availability of vaccines are by far the most important catalyst for our economy and for global economies to recover. I cannot think of anyone more qualified to comment on these issues on COVID-19 and the launch of the two vaccines announced in November than Dr. Keyvan Mojarrad. Prior to his current role at Walter Reed, Dr. Mojarrad was the Manager for Emerging Infectious Diseases at the Department of Defense, the head of the Joint West Africa Research Group, and the lead for the US Army's Zika vaccine and coronavirus programs. In his current position as Director of Emerging Infectious Diseases Program at Walter Reed, he took the group from a small unit without funding to a department with a $75 million budget. In 2015, he was named an advisor to the World Health Organization and advised on vaccines for Ebola and other coronaviruses. Prior to the WHO, he led preclinical initiatives at the National Institute of Health, where he focused on pathogens such as HIV, RSV, influenza, and MERS. He holds an undergraduate degree from Duke University and an MD, Masters of Public Health, and PhD from the University of Alabama. Without further delay, it's my pleasure to turn the program over to Dawn Brock. And Dawn, please take it from here.
Thank you, Dan, and welcome everyone. Dr. Majarid, first and foremost, thank you. You have dedicated decades of your life to these complex fields of medicine and science, and we are all honored and incredibly excited to hear your thoughts and your insights. As Dan said, this discussion could not be more timely. On one hand, we have COVID-19 infection rates at record highs, and yet on the other, we had this beam of hope this week as the first vaccine came to market. You are right in the middle of all of it as an epidemiologist, as a leading researcher, as a developer. So why don't we start there? You have worked with many pathogens in your career, but your work has been focused specifically on coronaviruses for some time. Can you talk about your experience and establish some context for us around the source of this novel coronavirus, SARS-CoV-2, and why it's so unique?
Well, thanks, Dawn, for having me on this forum tonight, and thanks to Dan for the very kind introduction. And it's a pleasure and an honor to speak to your audience. And thanks to my brother-in-law as well, Jason Shaw, for making the connection so that I could be with you here with you tonight to speak to you a little bit about COVID-19, coronaviruses, and the vaccine landscape, as was mentioned, as we try to end this pandemic. Coronaviruses in the history of pathogens is a fairly new pathogen. We learned about the existence of coronaviruses in the 1930s, first in chickens, and then we didn't really understand that they caused any kind of disease in humans until the 1960s. Until SARS-1 back in 2002 and 2003, there were only 2 known human coronaviruses that caused the common cold. Since then, there have been 5 new coronaviruses, including SARS-CoV-2. So part of it is that we have developed better detection methods to find these members of this coronavirus family. And it's also that we are accelerating the interface that we have with animal populations that have resulted in the emergence of more pathogenic, more deadly coronaviruses like SARS, like MERS, and now SARS-2. We saw this virus back in December. There was a strange syndrome that was occurring that was causing a severe pneumonia that looked very much like the patterns that were emerging during the SARS-1 outbreak in southeastern China. And we all had a sense that those of us who were working on coronaviruses, that this would be another new coronavirus, a novel coronavirus. We had no real indication that it was going to be more severe than SARS-1. Those of us working in this field, and I've been working in coronaviruses for about 8 years, I have been presenting a slide for the past 5 years in which I show SARS-1 in 2002, MERS in 2012, and then a coronavirus X in the next 5 to 10 years. And there are a number of others in my field who felt that another one was coming. We didn't anticipate exactly when that was going to be and how severe it was going to be. Clearly, the world was a little bit surprised that something as severe as SARS-2 and something as transmissible as this virus emerged. We all thought the next big one was going to be some variant of influenza. You asked about where this came from. All indications are that this has come from the definitive source, which almost all coronaviruses that we know of come from, is in bat populations, and usually fruit bat populations. Bats have this unique feature that they're mammals, so they're closely related to us, relatively speaking, in the animal kingdom, but also they have a lot of interaction with many different ecosystems. So they become sort of a melting pot for different pathogens that they collect from other species. And then usually there is an intermediate animal source that they pass it on to that then comes over to us. We haven't definitively identified that intermediate reservoir like we did for SARS-1, which were civets or cats, and MERS, which were camels. But a lot of groups are actively engaged in still trying to identify that definitive source.
So maybe from there, you can give us a little bit of context for the high transmission rate of SARS-CoV-2. So, the common comparator has been, the flu, which you have already said is really not the right comparator. So can you give us a little bit of background on why this virus is causing such high transmission rates and the severity of the symptoms?
Sure. So if we start with, there's a lot of still, what we have to understand is that it's still not even been a year that we knew of the existence of this new virus. So this time last year, there were 0 people working on SARS-CoV-2 because we didn't know the virus existed. So there's still a lot of questions, despite the tremendous amount of knowledge that has been gained about this virus within a year. There's still a lot of questions out there about the epidemiology, about the pathogenesis, about the immunology of this virus. Having said that, we're starting to get some hints, some clues as to why this virus is more transmissible than other coronaviruses, and particularly more than other pathogenic coronaviruses like MERS and SARS, the original SARS. I think the public has now been kind of educated as to this number called, the R naught, or the reproduction reproduction rate. And that is a number that tells you how many new infections will one infection produce. So if I get infected, how many more people will I infect if I come in contact with them? Anything greater than 1 means that you're going to propagate transmission and you're going to have an epidemic. And an epidemic doesn't stop until that number comes down to less than 1. So MERS, for example, doesn't transmit easily between humans. That has a reproduction number around 0.67, 0.7. Influenza, depending on the year, has a reproduction number around 1.1 to 1.5. The highest ones that we know of are something like measles. And this is important for later discussion if we talk about it, as to what kind of vaccines efficacy and coverage you need to be able to interrupt transmission of these very infectious or contagious diseases. But measles has a reproduction of close to 20. So one person could potentially then infect 20 people, and then those 20, another 20 each, it becomes exponential after that. SARS-CoV-2, by all estimates, there have been a lot of modeling estimates, a lot of epidemiologic estimates, but it's converged around a reproductive number of around 3 when it's at its worst. And the hypotheses as to why it has a double the contagiousness of flu and more contagious than SARS-1 is a couple of reasons. One is that it causes a lot of asymptomatic infections. So a lot of people who get the virus don't have symptoms or have very mild symptoms, yet they still have a lot of virus replicating in their upper airways and their sinuses and their noses. And so that makes them efficient transmitters because they're not bedbound, they're not at home, they're not in the hospital, so they can be moving out and about, not knowing that they have the virus and still being a reservoir, a source for transmitting the virus. Why does that happen? It's not entirely clear why people have such high virus loads yet don't have any symptoms. That is, I think, the key question that's going to have to be answered is why there are certain subpopulations that get so sick, whereas others don't get sick at all. The other thing about this virus is that compared to SARS-1, the part of the virus that hooks on to your lung cells that we call the spike protein, and particularly the part that binds the receptor in your lungs, called the receptor binding domain, is very sticky. It seems to be stickier than SARS-1 in terms of latching onto your lung cells. So that makes it, again, more likely to infect because it latches on a lot more strongly than SARS-1. And the other is that people who have with SARS-1, people who had the virus were oftentimes a lot sicker, as I mentioned, so they were not really out circulating in the community and transmitting the virus. Again, these are all hypotheses that are starting to gain some traction and data to support them, but we have not really definitively concluded the reasons just yet.
Got you. Look, that was an excellent explanation and foundation and segue for us to talk about the vaccine landscape, as you mentioned. There are several hundred vaccine candidates registered globally today, and yet they're quite differentiated by their platform technology, by their dosing regimen. Maybe you can help simplify the differences between the vaccines in this first round, Pfizer, Moderna, AstraZeneca, J&J, maybe Novavax, and their respective approaches, which really are different.
Yeah, so I think the best way to think about this first is that despite the hundreds of vaccines that are registered through the WHO, there are still scores that are in clinical trials, and then there might be a couple dozen that will actually, have any chance of reaching market. And all of those vaccine candidates, all of those different platforms come down to the same principle. The principle of a vaccine is to train your immune system, to educate it, to develop a response to the virus without actually seeing an infection. So that's the idea, is you don't want to get disease. You can get immunity from getting disease, but there's a chance that you'll get very sick. There's a chance that you'll die. There's a chance, of course, that you'll transmit to others. So why not develop that immunity without actually having to get the disease? And all these vaccines, again, come down to the same principle of delivering the part of the virus that we think induces the strongest immune response. And it's that hook that latches onto your lungs, that spike protein. If you develop an immune response, and one of the ways that we develop immune response is with these proteins called antibodies. If you develop the antibodies against that spike protein before you get infected, then when you are exposed to the virus, you already have these antibodies ready to go. And they attack the virus and prevent it from latching onto your lung cells and infecting your lung. So they all deliver that spike protein, but they deliver it in different ways. And that's what differentiates these different vaccines, these different platform approaches. And you can break it down into about 5 major buckets. You have the Moderna and Pfizer BioNTech vaccines, which are genetic vaccines. So you take the genetic code for the spike protein, not the spike protein itself. You take the blueprints for that protein and you inject the blueprints into your body. And then your body takes those blueprints and it makes the protein. So now it's made the protein and now it can present to the immune system. DNA vaccines are like that as well. But mRNA, messenger RNA, like the Pfizer and Moderna vaccines, have a little bit of an advantage over DNA in that they get into the cell better, they express the protein a little bit better. Then you have the vector vaccines. So you take, it's a different delivery platform. So you have the truck and it's just a different kind of truck in delivering, the payload. Here, the viral vector vaccines are the ones like Johnson & Johnson and AstraZeneca. And you take a virus, you genetically modify the virus so it doesn't cause any kind of infection on its own or any disease, a different kind of virus, not a SARS virus. So most commonly, and the ones that are used by Johnson & Johnson and AstraZeneca are adenoviruses. These adenoviruses are modified in a way where they don't cause disease, they don't replicate, but they infect your cells and again, deliver that payload, the genetic code for the spike protein. Then you have protein vaccines, where you're not delivering the genetic code, but you're actually delivering the protein, the spike protein itself. So you're taking the spike from viruses, you're modifying it a little bit to make sure that it's stable, and then you're giving that protein to the person, and then they are educated their immune system is educated to develop an immune response to that protein. And then finally, what is not really being taken forward in the US, but is one of the main platforms for a couple of Chinese companies, is a purified inactivated virus. So take the virus itself, kill it with a chemical method, and then use that killed version of the virus as your vaccine. There is a 5th category that is not being advanced so much, that is the same kind of way that we developed measles, mumps, rubella vaccines. Those are live attenuated viruses. So you're not killing the virus, you're just weakening the virus. That takes a lot longer to develop because you got to make sure that you got to be absolutely sure that that virus is not going to mutate in a way in the body that it'll revert back to the non-weakened form and actually cause disease. So it's a longer development process for that. Although those kinds of vaccines induce a very long-term type of immunity. Some of our best vaccines are these attenuated viruses. So if that actually comes to market, that won't be for a while. So those are sort of the main buckets, the main categories in which you can frame your understanding of how these different approaches differ from one another.
Okay. And you mentioned speed and how these different approaches, take different amounts of time. And there are a lot of concerns about speed to market and safety. And particularly considering the fact that we know that pre-COVID, the typical timeline, as you said, from lab to life was 5 to 10 years. So what are your thoughts around that? Has the technology caught up to the point where that was the gating factor for the timeline to be so long? Or were there a couple of different things that kind of amalgamated to actually push these projects and these campaigns forward?
Sure. As in many things in life, these things are multi-layered, and there wasn't just one driving force behind the speed at which we developed and got to some form of authorization for these vaccines. Um, when people say that it's taken less than a year, what I try to remind people is that it took less than a year from the time that we got the sequences of this new novel coronavirus to an authorization from the FDA. But it's been probably about 7-8 years that the technologies that have enabled these vaccines to go forward have been in development. And there are two parts to it. So if you look at mRNA vaccines in particular, since these are the first two that have gotten or will likely get authorization, there's been a lot of work done in the past decade on optimizing the mRNA platform. And doing it in the context of other viruses. Had we not had that data on these platforms for other viruses, we would not have been able to accelerate them now for SARS-CoV-2. The other part of this is the spike protein that I mentioned that these vaccines deliver. I told you that they're engineered in a way to make them more stable, to make them more potent in inducing an immune response. That genetic engineering of the spike protein was a lot of trial and error that took, again, about 5 years and even longer. Since my time at the NIH, about 6-7 years ago, we had just started working on that modifications to the spike protein. And only in the past couple of years, the past year really, did the NIH get it to where they could then translate it pretty quickly over to SARS-CoV-2, the kind of genetic mutations you had to introduce to get the spike protein in the right shape that would deliver a strong immune response. You're seeing the product, the sort of the fruits of labor of almost a decade of investments and research. And we caught a lucky break with this one. Had that kind of work not been done on other coronaviruses, on these platforms for the past 5 to 10 years, we would not have been able to go as quickly as we have now. One other point is that when we talk about the acceleration, there is no part of this process that has accelerated the safety review and the safety checks. All of those have been very religiously adhered to. Where the speed has come, and we always knew, those of us in the vaccine field, we always knew this 5 to 10 years could go a lot more quickly. It's just that things are done in sequence. No company is gonna put up the resources, put up the funding to manufacture product, to set up the infrastructure for clinical trials, even before you show that it works in animals. I mean, that's a huge risk. So that's why Operation Warp Speed and the US government stepped in to take on that financial risk. You know, Moderna received probably about $2.5 billion from the US government to help with the manufacturing so that there was stockpiled doses for when this vaccine gets authorized. So the next day or 2 days later, people can get started getting vaccinated. To set up the infrastructure at over 200 sites in the country while we were still evaluating this vaccine in monkeys and mice. If they had failed at those early stages, all of those investments would have been wasted, but the US government would have taken the hit, taken the loss. Industry is not going to take that kind of risk. And that's how you get to warp speed, by someone else stepping in and taking that risk at the manufacturing stage and at the setting up of infrastructure along the pipeline, but still maintaining all those safety checks. So the year-long process, actually, one is limited by the science, but we were able to accelerate that by having done science beforehand to some extent. and the other year-long process is the safety. Everything else can go pretty quickly if you have the investments in place to do things in parallel.
Look, I mean, I think that that's a phenomenal frame of reference, because I do think that there probably is miseducation out there just in terms of the timeline, and not that anybody could actually know all of the funding and all of the work that has been done, behind the scenes, excuse me, and over the course of many years, not necessarily in preparation for this, but in preparation for the advancement. And clearly the technology has caught up in such a way that there isn't as much of a disconnect between the speed and the safety. And I think that's important for people to know. The one vaccine that we haven't talked about yet is yours. You are leading the DoD coronavirus vaccine research project. And your group is using a novel approach with a unique goal, from my perspective, to develop, for lack of better terms, a broad spectrum solution with the coronavirus vaccine. So can you talk about your work and how far along you are?
Sure. Thanks for mentioning it. It is actually an Operation Warp Speed vaccine that is being tracked by Operation and supported by Operation Warp Speed, but it is obviously a second tier candidate and a more long-term candidate. And this fits in with the philosophy of my program. When I founded it, I wanted to get away from one drug, one bug solutions, so that we're not constantly in this reactive posture where an outbreak occurs and then we develop a vaccine for it and then pat ourselves on the back for going faster than we did the last time around. Clearly, the timelines that we have met for SARS-CoV-2 are far faster than anything done before, and are truly unprecedented. I mean, that word gets thrown around a lot, but it's true, it is unprecedented. But even though we got vaccines to authorization in less than a year, just under a year, 300,000 people in the US have perished. 1.5 million people have perished globally. Tens of millions of people. And by the time people actually get access to the vaccine who need it, there'll be many more that succumb to the disease that this virus causes, and many more that will have long-term health problems as a result of the infection and the disease despite surviving it. That's not fast enough. As fast as we go, it is not fast enough to start the process after the virus emerges. So our approach with other viruses as well, and particularly with coronaviruses, are to develop the vaccines for these pathogens before they cause the outbreaks, before we even know about them, before we even know about their existence. And you might say, how is that possible? Are we trying to predict? There's one part of this where you can try to do surveillance and look at animal populations and try to get a sense as to what's going to come out next. That is, I think we should continue to do that kind of work, but we also have the technology, the tools available now to develop vaccines that don't have to predict what's coming, but actually protect against all of them at once. And that's the approach we're taking. And we're taking it with a nanoparticle approach, which is a little bit different than Novavax's nanoparticle, in that we can actually put different parts of, or we can put spike proteins from, say, if we're talking about coronaviruses, we take spike proteins from different coronaviruses and we attach them to this nanoparticle, where its core is a protein that is ubiquitous in all human life. It's called ferritin. It carries iron, one of the essential elements of life in our bodies. But it has a unique property. It assembles into this very nicely ordered, symmetric ball, and you can attach your spike protein to that ball so that the whole ball is decorated with different spike proteins from different coronaviruses, and it looks like a virus, but it's a mosaic virus where it has lots of different spikes from all these different coronaviruses, so the immune system develops a very broad response. And the idea is also that there's a lot of commonalities between these coronaviruses. So the response, even though it's broad, hones in on those common parts of the spike protein across the entire family to elicit this response that whatever coronavirus comes, your immune system will be prepared for it. It is sort of the stuff that we thought might be of science fiction, but really, again, it is science fact. We have the tools to be able to do this. It just requires the investment. And I think we're now at a watershed moment in R&D for these potentially pandemic pathogens, where people realize that we need to be prepared before the outbreaks occur. And we've had very, very good data. One of the things that I'm doing now is actually writing up all those data to publish, and we'll start our clinical trial just after the new year. Again, it is focused on SARS-CoV-2, but it's also focused on other coronaviruses. So we're taking this long-term approach where we have a product that provides long-term protection, but also provides broad protection.
I mean, look, it's fascinating. I mean, because if you think about it, all of the vaccines being worked on today are really based on the initial sequencing of the virus, right? So based on its characteristics, and this is a good question. I mean, how would we expect it to mutate over time? And, you know, does a solution like using ferritin nanoparticles actually address that mutation, because you have kind of this 3D approach by attaching lots of different proteins?
Yeah, so people are concerned about mutations. There have been, little stories popping up in the news of mutations that have been emerging in the virus, but that's what viruses do. That's one of the reasons why we have to, if this is a foot race with the virus, we have to already be running, and we have to give ourselves a head start, because if we're starting at the same point and the gun goes off, the virus is going to be outrun us every single time. And one of the reasons is because they've evolved thousands of years to constantly be mutating to evade our immune system. Fortunately, coronaviruses are a little bit different than things like flu, where they don't mutate as much, or they have a better editing function. So all these viruses and all life, when you make copies of your DNA or RNA, in the case of these viruses, as you make copies of it, there's a little editor that goes along. It's like a spell check to make sure that you didn't misspell anything along the way. Coronaviruses actually have a pretty good spell check. Flu does not. And so coronaviruses don't mutate as rapidly. And the mutations that we've seen, there have been some mutations that look like it's made the virus a little bit more transmissible, a little bit more contagious, but it has not rendered the virus resistant to the vaccines we've made or the treatments that we've made so far. But we have to track those very closely. It's another reason, as you say, why we have to develop these broad approaches, so that we're anticipating not just different species, but different strains that might evolve within a particular species.
Okay. Oh, and before we move on to a different discussion, really the concerns in society and really all of the questions that we fielded from clients really revolved around who should take the virus, which one is the right one to take, how are people going to know who have allergies, or I think everyone's pretty well educated right now on comorbidities, but what about the one-offs? What about the, grandmother who gets vaccinated but has a daughter, who's pregnant and, and isn't sure if she can still transmit the virus.
Mhm.
So it just feels as though there's just a lot of uncertainty about which one for who. Do you think that we're going to get any clarity around that? Or do you think this is just crisis situation? If you fit into these buckets, you should be taking whatever you can get.
Well, normally the normal process is that um, you don't start vaccinating people 2 days after something gets authorization because you don't have the vaccines to do so. Um, and um, but we went through the same process that we do with other vaccines. So we went through the FDA review. Uh, the FDA, it met criteria for emergency use authorization. So it's not full licensure. It means that this is still rolled out and that data are still collected. It's sort of like even though the vaccine is being rolled out, it's sort of like a very large study that's still being done, so that we're still collecting data on any kind of side effects. The ACIP of the CDC also met, which is, again, something that's done in a normal process to decide who should the vaccine be indicated for initially. What are the sort of the top tiers, the prioritization of the vaccines? We do that for other vaccines that are licensed as well. And it's a risk benefit calculation. I think that's what we need to communicate. I think the scientific community has done amazing things, and we've sort of met the task, and now we rely on our communicators to message this correctly, to educate the public. And we as scientists, the onus is on us also to educate people about the science, but then our policymakers, those in communications who are sort of take the messages forward, then have to communicate the policy and the risks and the benefits of these different vaccines. I think one thing that is important for people to understand is we should take first the lessons from other vaccine campaigns and apply them here. So one example is, explaining to people that you cannot get the virus, you cannot get the disease from the vaccines. It might seem obvious to some people, but I think that is, square one. You have to make people understand that there is no way possible that you can get the virus and the disease from these vaccines, because none of these are the live attenuated virus. If it was live attenuated, there might be a very, very small chance, but none of them are. So that's number one. And then number two is, look, thousands of people in the US alone are dying every day now from this vaccine, from this virus. And hundreds of thousands are getting infected every day. So what are the risks of somebody who has heart disease, pulmonary disease, diabetes, is over 65, is in a long-term care facility? Risk is pretty high that they're going to get infected, and if they get infected, that they'll get pretty sick or die. The risk of getting a bad reaction to the vaccine, well, up until the vaccine was authorized in the UK, about 45,000 people had gotten the vaccine, and there were no major safety problems with that. Now, is there something that is 1 in 100,000 or 1 in a million chance or 1 in 10 million chance of a bad reaction? That's possible. We don't know because we haven't looked in that large population yet. That's why this is being given an emergency use authorization and not full licensure, because we still want to do a lot of close monitoring of people who get vaccinated to look for those very rare events. But if you compare those rare events to the much higher risk of infection, passing on disease to others once you're infected, and causing infection someone where they might get very bad disease and might die, then you start to see we have to do a better job of trying to communicate those denominators and those risk benefit calculations to the public that sometimes is not so obvious when people try to digest this information on their own. As far as the different vaccines come into play, that's going to, I think, evolve over time as more of these vaccines become available. Right now, our goal is trying to just get as many people vaccinated as possible with the vaccines that we have available, because we don't even have enough doses of those vaccines to vaccinate even enough of the population that is at high risk. So let's get there first. And then as new vaccines come into the landscape, we can start. And again, there are going to be more studies that are going to be done, because there may be situations where people get one dose of one and one dose of the other just because of logistics, preferences, whatever. And so we actually need to do some of those studies to make sure that there are no problems that emerge where you get one versus another, and that it still is effective when you do one versus another. And that requires a partnership, collaboration, and communication between companies in a way that hasn't been done before, and that's already happening.
Right. And just lastly, when it comes to kids, right now, I don't believe that there are any clinical trials that are addressing anyone under 16. Do you know of any? And how do you look at the childhood population being vaccinated?
Yeah, so that is an important question. Also, the question that you raised about pregnant women, because it's not been studied in pregnant women either. And we have to, be cautious in, the good thing is that there are data with these platforms in pregnant women, but obviously not with this particular virus. So we have to be cautious in how we advise women of childbearing age about getting these vaccines. But then for children as well, children get infected, but overwhelmingly they are either asymptomatic or mildly symptomatic. Now, we weren't sure for a while if they were large reservoirs for transmission, but more and more data are coming out that children going to schools does not seem to be a major source for the spread of this virus. So then it becomes a question of how important is it to vaccinate kids in terms of protecting them from disease and from interrupting the transmission of this virus throughout the population. And it looks like, there are other groups where it's more important right now. They're at higher risk of developing disease and death, and they may be more likely to pass on the virus from person to person. What we're going to have to do, we haven't done these trials yet. Usually, and this is again the normal process, unless it's a vaccine that's specifically indicated for children. But if it's not something that's specifically indicated for children, but it's for the whole general population, kids have different immune systems. And we have to design our studies a little bit differently for kids and measure things a little bit differently in kids. So what we try to do is test them in healthy adults first, and then a broader population, cross-section of adults, and then de-escalate in terms of the age as to where we test these. These studies are going to be done, they have to be done, but at the same time, we have to vaccinate our most vulnerable populations while we're doing these other studies.
Absolutely. Absolutely. Um, maybe we can shift gears a little bit and talk about the vaccine and the market, both the stock market and industry. You know, as we've touched on this interplay between industry and academia and government. You know, our audience tonight is a group of very educated, very sophisticated, and very enthusiastic investors. So we definitely need to get some industry talk in here. And you know, I want to start with tech. Dan mentioned it in his intro. You recently wrote a book, I think, on emerging vaccine technologies. I was hoping maybe you could share some of your thoughts on the recent advancements and what kinds of kind of tools and software of the future will help drive even more innovation. So, you know, where are the bottlenecks today that could actually enable us to replicate the effort that we just put forward on the vaccine front for COVID-19?
Well, thanks for putting in a plug for my book. It's a couple of years old now, but I think some of the lessons still apply here. And amazingly, sort of what's the content of that book is what I thought would be something that wouldn't happen for decades, has happened over the past year, and it is taking these different platform approaches that I mentioned, and finding ways to to have them in place, um, with kind of what we call conserved parts of these different viruses within families, um, and inserting those into these platforms and testing them. Some of these things were already starting. So this is a watershed moment, but there was a watershed moment just only 5 years ago, I think, for global health and for global development of vaccines, and that was the Ebola outbreak of 2014 in West Africa, and that spurred the World Health Organization to do some introspection, and they were charged by the UN General Assembly and the World Health Assembly to do a after action report analysis of how they responded to some extent, not optimally to the West Africa outbreak, and find ways to, at a global governing level, how to bring industry and the public sector together to work on these issues and these pathogens and set up systems for public-private partnerships to kind of hit the ground running when these outbreaks occur. And that also then spurred on something called CEPI, the Coalition for Epidemic Preparedness Innovation, CEPI, that kind of spun out of this. And that was already starting this process of investing, and the funding came from different national governments and from philanthropic organizations like Gates Foundation and the Wellcome Trust, as well as the governments in Norway, Japan, India, and others, to start generating data, clinical trial data, human data on vaccines for pathogens that we think are going to be the next pathogen of pandemic potential. But that's where I say, in our book and in the platform that I'm taking forward, and every time I have a forum like this or on media, that's not enough. It's not, because we're not going to see these things occur exactly the way that it had before. We're not going to see Ebola come out the same way, or another coronavirus come out the same way. It's not going to be the same one, but we have a pretty good idea that it's going to be one of a handful of families of viruses. All these different viruses that have been causing these outbreaks fall back into the same categories of about 6 or 7 families of viruses. So If we focus on those with the technologies that we have in developing the platforms and the vaccines targeted to those families and not individual viruses within those families, we'll be much better prepared. There is also the other part of this that is not just kind of the basic science, but is also the how we do business. And there has to be ways to incentivize industry to take part at some level in this. So it's going to require the public sector, it's going to require governments, philanthropic organizations to de-risk the process for industry, to make it attractive for them to develop these kinds of vaccines. We can't do it without industry, especially here in the US and most of Western Europe, Canada, and other places still have nationalized vaccine programs. We're going to need the support and the buy-in from industry to be able to do this. And that's something I think is a conversational process that started after the Ebola outbreak, but clearly is going to have to mature a lot more in the wake of this pandemic. And Operation Warp Speed is something that we can learn from. It was a very expensive endeavor. So clearly we can't do this for every single virus, but there are certain lessons to learn as to how we got industry to work together, with the government being the arbiter of different industry partners, and then finding a common path forward where you respected intellectual property rights, but also made sure that you got the vaccine to the people who needed them most. That's going to be kind of a very, I think, complex navigation process going forward, but I think we've made, major strides in the past year in that part of it as well. So it's the science, and then it's also sort of the politics and business of how these companies and how these public-private partnerships evolve.
Right. Well, and taking it, to industry and to the market, and maybe taking technology, but moving past the R&D stage. The funding that's gone to R&D has been fantastic, but there's still an entire bioprocessing, manufacturing, and logistics supply chain that's behind the actual product getting safely into our arms. So, it's been interesting to watch the market because there has been so much strength around these associated areas. They didn't necessarily get funding or need funding, but technology has played a huge role. And that's everything from manufacturing and consumables to transportation and logistics, to the diagnostic players. Obviously we're going to have a lot of testing going on, but investors think that there is a multi-year money flow opportunity here that's going to support this treatment and cure. What do you think? I mean, do you think that we're going to get this right quickly, or most of it right quickly? Or are we going to be looking at this and really investing in it and, take this as a learning experience and something that can inform the way that we look forward so that maybe we don't endure another pandemic, maybe it's an epidemic?
Yeah. Well, first, I'm a scientist and a physician, and I want to start with that qualification because I chose to pursue that path in life and not business. And so, I want to tread lightly when I talk about how the markets will respond in the future and how they'll evolve in this space. Second, I wanted to say that, you brought up a good point that in our field, we kind of have this saying that, vaccines don't matter. Vaccinations are what matter. And I would say even more so, immunizations are what matter. So that if we do all this work and get a vaccine on the shelf and no one takes it, then what was the point? So, and we're talking about this in the context of vaccines, but you can apply this to other products in the healthcare space, devices and drugs and diagnostics as well. But it's going to take a lot more than just investing in the basic science. I understand that part of it. But I think it's going to take a change in mindset, a change in our culture, to not focus on what's just in the spotlight. I have seen this too many times in my short career so far, that after an outbreak occurs, after an epidemic, there's lots of money pumped in, lots of attention, lots of discussions that go on, and then it fades, and then we sort of forget about it, and we don't make those foundational investments into not just the science, but the infrastructure, the logistics, the supply chains, as you said, the consumables. I mean, there was a huge problem early on in this years in just having the basic materials for the R&D process for and the deployment of these different products, from diagnostics to drugs to vaccines. So that is a multi-year process that has to take place. And I guess, that does present an opportunity, because it will involve many sectors of industry, of the market. But we just have to, one example I like to give is Zika. So we had, Dan mentioned in his introduction, I led the Army Zika vaccine program. And we got to a point where we were partnering with industry to take this forward, but then the epidemiology changed. Well, I continued, and then nothing happened, and everybody stopped doing Zika. But our program is still doing the work to develop the Zika vaccine, and so that we're ready to take this forward once it comes back. And it will come back, because new populations will emerge. The mosquito will spread, it'll enter into populations that hasn't entered. And we have to be ready for that, meaning before it actually happens, not after it happens. And I know I'm sounding like a broken record here, but I think if we can make that shift as a population in doing the kind of, instead of putting out the fires, doing the work for prevention of fires, then I think we'll get to the point where we'll avoid these pandemics, and we'll build a whole industry, a long-term industry around this to prevent these pandemics.
That was a great answer. In our last few minutes, I know we're bumping up on the hour, but I really do just want to look maybe 6 to 12 months ahead to, living in a post-vaccine development and distribution world. What do you see? Do you anticipate that there will be a new normal that includes, or maybe even requires, a permanent shift, right? So right now, it's socially acceptable. It's only socially acceptable to be distanced, to wear masks. And, is there a world, even 2 years from now, where the water fountains come back on in the schools, or there's a path to pre-COVID living in your mind?
First, I hope we don't return to the old normal.
I knew you were going to say that.
And I've already kind of more than hinted at that in terms of how we approach these pathogens, right? But I think this whole experience, stepping outside of my head as a physician and scientist, and as just a regular person, dealing with the same issues that all of us have had to deal with. I think it's made us understand what's important in a lot of ways, and truly, hopefully, value what is most important. So I hope we come back to a different kind of normal, but one in which, yes, we have water fountains and we eat in restaurants and you spend time with each other and have parties and all those things that we did just a year ago. Uh, when you look back to the Spanish flu pandemic of 1918, it was actually took several years,
Right.
and we didn't have vaccines then. We didn't know what viruses were then. Um, so even with that, we eventually got back to a society that's physically interacted with one another. So I think we'll get there. Um, I think it'll be important to do this carefully. I think because we still don't quite understand what it's going to take to get to that point where um, transmission is being interrupted, what sort of level of vaccine coverage we need to get to. So I think we should do this in a very rational and staged way, and not get too excited and well, the vaccine's here, let's start having concerts again with hundreds of thousands of people. We have to do this in a smart way, but I think we'll get there, but hopefully with a shift in mindset as to what we place emphasis on in our lives and also in R&D as well.
So herd immunity, I mean, just we'll end there. What does that mean to you? When do you think we reach it? What do you think has to happen in order for us to attain that level of comfort and protection?
In the short term, it means that there are enough people who are immune, whether by natural infection or vaccination, that the virus doesn't propagate efficiently, so that you drive that reproduction number down to less than 1. It's all about that. And for the long term, it's about making sure that either if you were infected or vaccinated, that your immunity lasts. And we don't know that yet. We don't know how long your immunity is going to last. If you look to other coronaviruses, the ones that cause a common cold, we know that people can get infected after 2 to 3 years again, although usually with a milder disease even. But we still don't know the answer to that question as to when people can get reinfected. If they do, what kind of symptoms will they have or not have? And so we have to make sure that we're not complacent. And do we need boosters then? How often do we need those boosters? And what are going to be the logistics of making sure that we're tracking people and bringing them back for boosters? It's hard enough to just bring them back for one boost.
Right.
So bringing them back several years later for another boost, that's going to be challenging as well. So again, that's going to require we have a very good system of doing that for kids. We don't have a very good system for doing that in adults. So herd immunity, I think we'll achieve in the short term fairly quickly, hopefully within the next year. But for the long term, that's still an open question.
Fair enough. Well, maybe we can, first of all, thank you. This has been an incredible conversation. We have learned a lot from you, and maybe we can reconvene. Maybe we can have a round 2, and as more information comes to light, and as we get closer to herd immunity, we can look to the way that industry and government and academia, continue to work together, hopefully, and hopefully a much safer outlook for all of us.
And hopefully we can do this in person one day.
That would be lovely. That would be lovely. I'm sure that Allison and Dan will be very happy to set that up.
Well, thank you. Thank you for this opportunity.
So very much. And we did have questions in the queue, but I think that you have done a phenomenal job of taking us through this current crisis. So thank you and have a wonderful evening.
Thanks very much. A good evening to all of you as well.
The information contained in this webcast reflects the views of Palisade Capital Management, LLC, as of the date of its original broadcast, and such views are subject to change without notice at any time. All information provided in this webcast is for informational purposes only and should not be considered investment advice or recommendation to purchase or sell any specific security. While the information presented herein, including information obtained from third parties, is believed to be reliable, no warranty is made concerning the accuracy of such information. There can be no guarantee that any forecast discussed in this webcast will be realized. Consult your financial advisor before making any financial planning or investment decisions. Past performance is not a guarantee of future results.